Welcome to Two Bees in a Podcast, brought to you by the Honeybee Research and Extension Laboratory at the University of Florida's Institute of Food and Agricultural Sciences. It is our goal to advance the understanding of honeybees and beekeeping, grow the beekeeping community, and improve the health of honeybees everywhere. In this podcast, you'll hear research updates, beekeeping management practices discussed, and advice on beekeeping from our resident experts, beekeepers, scientists, and other program guests. Join us for today's program, and thank you for listening to Two Bees in a Podcast. In this episode of Two Bees in a Podcast, we will be talking with Dr. David Tarpy, a professor from North Carolina State University. He is a world expert on what it means to have a quality queen. And furthermore, he'll be chatting with us about his queen and disease clinic run out of NC State. That will be followed by a segment where Amy and I discuss the biggest stressors that honeybee colonies face. And of course, we'll end today's podcast with our question and answer segment. Hello, everyone. In this segment, we're going to be talking with Dr. David Tarpy, who's a professor and extension apiculturist in the entomology and plant pathology department at North Carolina State University. David, thank you for joining us on Two Bees in a Podcast.
Thanks for having me, guys.
Yeah, it's really cool to have you on. I see that we're talking specifically about one of the programs that you run out of your lab at NC State, the Queen and Disease Clinic. But before we get there, David, I've known you for, I guess, about a decade now. So I'm curious if you could tell our listeners how you got into beekeeping, how you found yourself at NC State.
Well, I got into honeybees as a model system for the study of social behavior and behavioral ecology. They're kind of, the white lab rat for social insects because they're just so highly amenable to experimental manipulation and those kind of things, going all the way back to Carl von Frisch and his, Nobel-winning research on the dance language and everything in between. And so I've always been really fascinated at the collective decision-making and how colonies work together, and especially centered around queen biology. And so I got into it from that aspect. But beekeeping is part of, having to do research on honeybees. And so I love to tell beekeepers that I actually didn't read my first beekeeping book until about 7 years after I had been keeping bees, because I knew enough about the biology that the beekeeping was just kind of obvious, and it just made a lot of sense knowing how the bees, work themselves. And so once I took the position here at NC State in 2003, the extension mandate that comes as part of our program really fostered that communication and working together with beekeepers. And so the applied research and the beekeeping became a lot more prominent in what we did. But that wasn't how I started out.
Now, what is your academic history like? What universities did you pass through to get to NC State?
Oh, I got my undergrad at a small liberal arts college called Hobart in upstate New York, and then I got a master's degree.
What? Really? Me too. No, I'm just kidding.
Yeah,
I'm kidding.
Because you're not, because that's an all-male college.
You got to watch Amy. She tries to be funny.
That's an all-male college. The all-female college is called William Smith. So
together
they are Hobart and William Smith. And it only has 1,700 students. So and I've only met one person down here ever since. So I don't. I highly doubt that. But yeah, so I got my undergrad degree at a small liberal arts college called Hobart in upstate New York. And then I got a master's degree with my mentor, my academic mentor, Dave Fletcher at Bucknell University. And that's where I first got into honeybees, where we were studying the competitive bouts between virgin honeybee queens to see who takes over the nest. And I saw, actually, incidentally, that your last podcast dealt with the tooting and quacking between the virgin queens and that kind of communication. So that's actually kind of what got me into honeybee research. I then went from there to UC Davis, California Davis, with Rob Page, and got my PhD, and then a postdoc with Tom Seeley at Cornell before I got this job here at NC State.
That's really awesome. I love hearing about everyone's story and their background.
Well, just on a side note, Amy, as Jamie probably know, we're all inbred. So it's not too far to say, like, Jamie is probably my academic, like, second cousin or something like that, right? Like, I'm not quite sure how we're related. I'd have to think about it, but
sure
I know we are.
I feel like we need to make a family tree with all the podcast guests that we've had, Jamie.
I've always wanted to do that. If you guys are up for it, I'm totally down for it, because in fact, I've always wanted to write a book, 6 Degrees of Separation from Roger Morse. Because it all goes back through Roger Morse.
Honestly, I think, David, we are all probably 1 or 2 degrees from Roger Morse, because a lot of the individuals who are leading labs these days were in his lab, which is pretty fascinating to think about.
Well, now it's actually a second generation from that, right? It's
that's a good point.
So yeah, I mean, yeah. So anyway, sorry.
That's so
this is
awesome.
all to be edited out later, so I'm glad you guys are down for that. So you broke the seal there, Amy, with your joke. So.
I tend to do that. All right. So I guess one of the things that we like to tell all of our audiences are 3 main focuses when we're talking about research and honeybee research in general is varroa, queen quality, and nutrition. So those are the 3 things that we really kind of focus on. Of course, there are other factors, there are other stressors that we look at, but primarily those are the 3. And you have a queen and disease clinic at your lab, right? So can you tell us about the clinic that you have?
Yeah, so the clinic is really an outcome from a lot of our research. So we did a lot of the basal research of understanding what makes a good queen good and what makes a bad queen bad. And so trying to understand the bounds of that from the queen's perspective is where a lot of this started out, both their kind of physical prowess as well as their mating success and their mating quality. That then led to these questions about, well, does a good queen make a good colony, and does a bad queen make a bad colony? And so we did some of those studies and found that there is an association. It's not, an absolute 1-to-1 association, but there definitely is that better queens make better colonies. And so we then started offering this service in 2013 to bring to bear the same scientific techniques that we use in our research to inform beekeepers about the quality of their queens in much the same way that we do, in a very high throughput way. And so, beekeepers can send us queens. It's a destructive sampling, so unfortunately we can't send the queens back, but we can measure them in a very high throughput way for both their physical abilities as well as their insemination success, looking at sperm viability, sperm count, and those types of things that are invisible to the beekeeper but can have profound effects on the colonies. And then we send reports back to the beekeepers to say how good those queens were. And if, a beekeeper makes 1,000 queens and they send us 10, and we tell them, how they rated on the scale that we've developed through our research, then that will give them information about the other 990 that they can then turn around and sell to other beekeepers. So it's a way of providing some quality assurance of queens that are produced by queen breeders large and small. We also do a lot of research on pathogens and colony health. And so the other half of that clinic is the disease side, where we do pathogen screens for beekeepers, usually on the workers, to be able to look at other invisible or asymptomatic pathogens such as viruses.
So let me ask, David, there's two questions that come out before I even get to the schedule question I have for you. What are the qualities that you're looking for with a queen to make her good or bad, right, when she comes to you in the clinic? And also, how many pathogens are you screening for? Are they principally viruses, or they include bacteria as well? And I know that's 2 questions, but if you could address those, I'm curious.
And I will, and I'll repeat the questions. So what we look for with queen quality, we really break down into 2 general categories. That is the physical size of the queens, and then the mating success of the queens. And so the physical size are things like body weight and head width and thorax width, and some of the very kind of easy low-hanging fruit that we can measure using digital photographs very accurately. And larger queens tend to do better and tend to score better in our system. And that's, again, been borne out by the science. And then the other category, we dissect out their spermathecae, the sperm storage organ where the queens store their sperm for their lifetime. We dissect that and we stain the sperm, and we use an automatic cell counter to look at both live and dead sperm, so we can give sperm counts, sperm viability, and other measures dealing with reproduction. And so we can put both of those things together to provide an overall assessment of quality. So there's lots of measures that go within each of those, but those are the two general buckets that.
And you generate a score as like a 1 to 10. It's 100. I mean, what does the beekeeper receive in return?
So you can see examples of the reports that we give back at ncsuapiculture.net. But we're at the university, so we got to give out letter scores, right? So we actually have broken down this standard measuring stick that we know on the one end are good queens and on the other end are bad queens. And so we just give a letter grade, an overall letter grade, and then we give letter grades for both the size and then the insemination success. And then we actually give the raw data for each individual queens and each individual queen in case the person is interested in differences between different breeder stocks or different mating yards or those kind of things. And we're very willing to work with beekeepers to be able to troubleshoot or even engage in custom collaborations where we can answer very specific questions for them. So, for example, we've had beekeepers who are worried about, how long is too long to bank their queens and then be able to sell them, as high quality queens. So we've worked with them on that. We've worked in beekeepers to see, how early in the season is too early and how late in the season is too late. And so we've done kind of temporal analyses of their queens. We've looked at all different aspects, like looking at this new mite control product. Does that sterilize their drones? So there's all sorts of ways that this clinic can be brought to bear.
That's really interesting. I'm thinking of like a queen coming in and basically going to the equivalent of like a Planet Fitness to do, like a health analysis and coming out with a score and what their like body weight is, So
this one dies, Amy.
Well, okay. Some people feel like they want to die after that.
Well, I would love, we're definitely working towards some sort of non-destructive sampling. That would be ideal,
Sure.
But right now it's this kind of subsampling of a larger cohort is really the only way around it. We could do the non-destructive if the body size was a better proxy for the overall quality. Instead, the size does matter, but what really matters is that sperm count and some of those other measures that can only be attained through destructive sampling. So we'll work towards that. It would be nice to put them on a treadmill and just see how they are and then give them back to the beekeeper, but we're not quite at that point yet.
That's fair. Just real quickly, how long does it take from the time that beekeepers send in samples to when they receive information back?
Well, when we first started the clinic, it was something like, 2 to 3 weeks or even more, but now we have it down to a turnaround time of 1 to 2 days. Depending on
Wow.
the schedule and the number of other samples that we're doing at the same time. The main person who really runs the queen side of our Queen and Disease Clinic is Dr. Brad Metz, who does that. And then on the disease side, it's our genetics technician, Aaron McDermott. So beekeepers can work directly with them as far as shipping logistics and all of those other things. But usually on the queen side, it's a very quick turnaround, and it's very, very helpful, especially for larger commercial queen producers.
And the pathogens, what pathogens do you screen for? You don't have to name them, I guess. How many do you screen for, and how long does that take as well?
That takes a little bit longer because we're using quantitative PCR in order to diagnose those. Right now we screen for largely viruses, so acute bee paralysis, black queen cell virus, both the A and the B forms, the deformed wing virus, which is that calling card of Varroa, and it's really, really problematic and nearly ubiquitous, right? Israeli acute paralysis virus, like Sinai viruses, But then we also look at both nosema species as well as trypanosomes. So it isn't just the viruses per se, but we are looking at some of those other pathogens. Now, we're not looking at the brood diseases and those things, although Aaron is busily working on creating a second panel of potential pathogens for things like European foul brood, American foul brood, whatever this snot brood, issue is, sac brood virus and, chalk brood. So we're working on a second panel and the logistics for that. But right now we're just looking at those main economically important diseases that can be found in the workers of the colonies.
Sure. And so, David, do you see hobbyist beekeepers using this a lot, or is it principally for commercial beekeepers, or it services any beekeeper who's interested in knowing about their disease and pest load or their queen quality?
So we are open to anybody to use it, and we do argue that it is just as useful for hobbyist beekeepers or smaller scale beekeepers as the larger scale beekeepers. But because on the queen side it is destructive, the quality assurance part is not as amenable to somebody who's only raising 5 queens. It's hard to get a subsample, a meaningful subsample with smaller populations. But it is really useful to say, hey, this queen is kind of pooping out. I wonder if it's because she's running out of sperm or for some other reason. And so hobbyists can send in their samples in those ways, kind of in a troubleshooting way rather than in that quality assurance aspect. And then also, again, the viruses, these invisible but incredibly important harbingers of problems and health problems that beekeepers are having. Beekeepers large and small can use that for sure.
That's great. We're going to definitely add some of your resources onto our website, onto the additional notes, and so people can go to that. And so I guess my question is, do you take samples from beekeepers around the nation? How does someone get involved in this? And if you don't mind, can you tell us what the cost is for, the clinic? Because I know, if it's for me, if I'm a hobbyist beekeeper, I guess depending on how much I really love my bees, there's a certain threshold I'd be willing to pay to figure out what happened versus just starting fresh and starting new.
Yeah, so we do take samples from all across the nation. So it's not limited to just North Carolina at all. In fact, most of our beekeepers that we're working with are in fact out of state. And the cost for the standard reproductive quality assay and report is about the price of a queen. So it's actually $24 a queen or a drone. We've also added drones to the mix. And so, it's about the price of a queen that, again, unfortunately, you don't get back. For the pathogen screening, it's $55 a colony, but we also offer kind of a pooled sample for an entire apiary, where you can do up to 10 colonies for $220 if you're interested more at kind of the apiary level rather than individual colonies. And so those are actually very competitive for these processes. The genetic analyses obviously are a lot more expensive. And while the techniques that we use are fairly standard, nobody's able to put the queen quality measurements together, and especially in such a high throughput and fast turnaround way that we can through this clinic.
That's really neat, David. I'm really impressed specifically too about the price of the queen one. That's really neat. I think that a lot of people could take advantage of that. And as Amy mentioned early on, we hear a lot about queen quality. I mean, you see it in the BIP reports every year. In the top 5, people are talking about queen quality. And anytime people ask me about that, it's a little amorphous to talk about. Well, it may be brood pattern or not. I saw your paper come out, talking about brood patterns and predictors of queen quality. It may be size or not. And just yesterday, for example, I was speaking to a group on Zoom in Ohio, and they were asking me about size of queen versus quality. So, we hear people telling us queen quality matters, but then we're not sure how to tell people to improve that or how to assess that. So it's really neat that you guys are providing.
Jamie, that's an incredibly important point. I'm so glad that you brought that up, because what we are learning through this clinic is that beekeepers are really well trained to blame the queen even when it's not her fault.
Yes.
And so we have gotten, I would say, the majority of the queens that were sent in being accused of being trash not well mated, early laying, early drone laying, other of these queen problems.
So who should we accuse? Who would we blame?
So this is the difficult part, right? Like, it's very easy to blame the queens, but we often test these failing queens and they're perfectly fine. So that then opens up an entire can of worms that, well, maybe there's something with the management practices, maybe there's something other things that are going on. So it makes these queen events, these queen problems, much more complicated and many different routes to that same end. But we need to get away from that blame the queen first mentality, I think, as beekeepers, if we're really going to solve some of these problems. We're really finding that it's really the environment, the nest environment, the toxicological environment, the nutritional environment, many of those other things that you mentioned earlier can affect queens. So if you think about it from the colony's perspective, if they're being besieged by all of these other stressors, but there's not really much they can do about those things, kind of like us, right? Like, we can't do much about our government, but once every 4 years, we can vote the president out or not, right? And so it's the same kind of collective decision-making process that's going on for the queen, that, hey, there's something wrong with the colony, must be the queen, let's get rid of her. And then you have these early supersedure events. And so I think we need to collectively step back as beekeepers and stop blaming the queens in the individual sense, but really looking at the queen as a reservoir or as the receptacle from all of these other stressors that the bees can take it out on her. So if anything has really taught us anything from the clinic, it's been that.
It is pretty interesting to think about it. You're right. Worker bees can live 6 weeks, right, in the summer. They can live 6 months, et cetera. Drones can live their period of time. But queens, in theory, can live a year or longer. And so they're really the bee that's in that colony that has the maximum amount of exposure to everything that's in that hive. They'll have the maximum amount of exposure to pesticides. They'll have the maximum amount of exposure to diseases and pests because they live longer, right? And so all of that can manifest. It seems like what you're saying, if you set a pretty average queen in a colony that is otherwise doing well, you're setting her up to succeed, and a lot of problems will go away altogether. And it's really, again, it's neat that you offer this service. I think what we're going to do for sure is make sure we have links to all of this information that you've talked about in our show notes. You can find that at our website. Everyone listening, just go to our podcast on the website, check out show notes. You'll see all this, the links to this, the disease and pest clinic, et cetera. But David, before I let you go, I'll be a little bit remiss if I don't ask you just to talk very briefly about your job at NC State. You've talked great about this disease and pest clinic. That's fantastic. This queen clinic that you guys have up and running. But I know you guys are a land grant, which means you have responsibilities in research, teaching, and extension. So could you give us a very short overview of kind of what your program does in all three of these areas? You don't have to go into great detail. We'll have you back in the future to talk about these, but I want to make sure that our listeners are just intrigued to go to your website and visit it and check out what all you guys do.
Well, just like your program, Jamie, we pretty much have the exact same three-way split, right, of research, extension, and instruction. And just like you, we run our program where we don't have these silos of those things, that it's all encompassing and one and the same. The clinic is really a great example of that, where it's an extension initiative that we currently have, but it wouldn't exist without our research that went, a decade or more of research that went into that. And so it's really a continuum where these things feed on each other. We can't do extension without the research. The research has no meaning if we don't do any extension. And then the instruction, of course, is centered on all of that, both instruction in the traditional sense for the students on campus, but also instruction in the non-traditional sense that starts to bleed into extension. So I really see it as a circular continuum of all of those things, rather than these dichotomous or these trichotomous kind of siloed things that don't inform on each other. And so just like you guys, we see that in its entirety.
Well, David, thank you so much for joining us on Two Bees in a Podcast. Everything you've talked about is intriguing, and I'm sure our listeners are going to want to check out your website to find out more about what you do.
It was a pleasure. Keep up the good work.
Absolutely. Everybody, that was Dr. David Tarpy. He's a professor and extension apiculturist in the entomology and plant pathology department at North Carolina State University.
Have questions or comments? Don't forget to like and follow us on Facebook, Instagram, and Twitter at UF Honeybee Lab. All right, everyone, in this segment, Jamie and I just wanted to talk, and I mean, that's what we're here for, right? Is to just talk.
That's what podcasts are.
So we wanted to talk about the specialty that we have in the lab and all of the research that we kind of focus on in the lab. Just generally speaking, we're always talking about Varroa, nutrition, and queens. So in every single one of our segments, there's always something about one of those three. And so we have had a lot of questions, just practical questions from the audience on, what all of those things even involve. I mean, why do we care about Varroa? I feel like people generally know why we care about Varroa, but where did it come from? We kind of talked about that too. But, then we'll move on to nutrition a little bit, and then queens. Of course, for some reason, Jamie, have had a lot of questions about queens lately. People want to know about queen rearing, breeding.
Yeah, it's just one of those things that people really are gravitating towards. I think the more we talk about it, the more it incites people to ask more and want to hear more about it. So I think it's kind of a vicious cycle, Amy.
Yeah, definitely. Okay, so let's start with the first one, Varroa. We have news reporters that call us all the time asking about Varroa. We always talk about Varroa. So let's just start there. What's the deal?
Yeah, so Varroa they're a mite that came from Asia, and there's 9 different species of honeybees, and Varroa's natural host is one of those 9 species, and it's one of the species that lives in Asia. So the reason Varroa is such a problem for our bees is because when European-derived Apis mellifera were taken into Asia, Varroa got on them and then spread into Europe, Africa, North America, all around the world. So essentially, you take this mite that is not a big problem for its natural host, Apis cerana, move it to a new honeybee, Apis mellifera, the bee that we keep, and all of a sudden it just wreaks havoc. And that's because our honeybees don't have a lot of natural defenses against a mite that they, quote, didn't grow up with. And so now we have this mite that was introduced in North America, or at least found in North America in 1987. It had been in Europe longer, some other places around the world as well. And it came over and it just started killing colonies. And it's been the world's biggest honeybee-related problem since. And in addition to the damage that it does to bees, physically, it also has been shown to spread pathogens. So you've got this big old thing feeding on you. And furthermore, when it feeds on you, it's going to give you diseases. And I mean, this is like the perfect mess, right? And so Varroa has just been that thing that's so good at killing our bees. And I almost refer to it as the cancer of the bee world, where millions upon millions upon millions of dollars have been poured into trying to address Varroa, but we're only kind of taking these small, modest steps, and a lot of what we do just seems still, very brutal to try to address Varroa. So I'm sure we'll talk more about that, but it's just a big problem. It's a big problem everywhere that Apis mellifera, this Western honeybee, is kept.
Well, so you were talking about Apis cerana and Apis mellifera, and you said there were how many other species?
So there are 9 total species. Well, there's probably more, but at the moment we recognize formally 9, even though some of the research in our lab suggests that there may be more. You guys just need to stay tuned. But nevertheless, there's 9 recognized species. 8 of those occur in Asia. So the only honeybee that occurs naturally outside of Asia is the honeybee that we keep, which is not called the European honeybee, contrary to most popular myths. It's called the Western honeybee. It's distributed in Europe, the Middle East, and Africa. And the ones that we have in North America came from Europe, which is why people like to call it the European honeybee, but it's most appropriately called the Western honeybee. But anyway, Cerana is an Asian species of bee, and it's the natural host for Varroa. And then, of course, the rest is history once it made it to mellifera.
So does Varroa have an impact on the other species of the other Apis species?
Not really. It's funny that you asked that, Amy, because even on Cerana, its natural host, it obviously is reproducing, it's feeding on Cerana, et cetera. But Cerana has a few behavioral mechanisms, grooming. It has some brood development issues that help kind of combat Varroa. But I've just not read a lot of papers about the movement of Varroa to these other Apis species and the impacts that it has. I mean, the biggest impact that Varroa has seems to be on mellifera, and it's possibly because that it developed away from mellifera, just significantly away.
So you were talking about feeding. What does the Varroa feed on? I mean, how are they doing damage? You said they're doing damage, but how are they actually doing damage?
That's a great question. So ever since people have been aware of Varroa, people have believed that Varroa has fed on honeybee hemolymph. So hemolymph is just a fancy word for insect blood. So honeybees have hemolymph or blood. And it kind of
like what mosquitoes would do.
Yeah. But it also makes sense when you look at a Varroa. It's affixed or adhering to the bee a lot like a tick would on us. Sure. Right? Ticks, if you walk through the forest, ticks like to kind of crawl up and kind of get in those little tight spaces, around the clotheslines and things like that, and they'll embed and they'll suck your blood. And so when you see Varroa doing something similar, you're like, oh gosh, yeah, that's what they're doing. They're getting on bees and they're feeding on bee blood. And we just kind of perpetuated this idea in the literature while we talk about it, just in our belief system, that Varroa feed on bee blood. That's just what they do. Last year in 2019, a team of scientists from the University of Maryland, the USDA in the Beltsville, Maryland area, my team here at the University of Florida, we collectively published a paper showing that Varroa, in fact, feed on fat bodies. And fat bodies are these lipid tissues that are around the bees that have a lot of different functions, pesticide detoxification, nutrient handling, et cetera. We could go on for days. But what we now know is that Varroa feed on these fat bodies. And when they're removing these fat bodies from adult honeybees, there can be this cascade of impacts that we see with honeybees that more appropriately match what you would expect if fat body was the food resource rather than hemolymph. And it's funny because Alan Cohen, who's a specialist with mites, he is the one who suggested that this was the case in the first place. He looked at Varroa morphology. He looked at the mouth parts. He looked at excretia, the poop, and suggested that all of these things don't point to a liquid diet, which is what you would expect if they were feeding on hemolymph, but instead kind of point to a more solid diet, which is what you would expect if they feed on fat bodies. So the abbreviated history of that system is that Alan told Jerry Hayes, who's a former colleague of mine here in Florida. He's now moved on. He's the editor of Bee Culture. Jerry Hayes told me and Dennis Van Engelsdorp, who's a professor at the University of Maryland. And very quickly, that project trickled down to one of Dennis's PhD students at the time, Sammy Ramsey. And collectively, we worked with, all of us worked with Sammy to help kind of put the pieces together. And Sammy published that paper as lead author talking about Varroa feeding on honeybee fat bodies. And it's already made a huge impact literally around the world. It's probably the most significant Varroa discovery in the last 2 or 3 decades.
That's pretty cool. So is there a possibility that there's anything else that they're feeding off of?
So you have to know, right? If something is piercing an insect, it's likely not just fat bodies that are getting into the Varroa. I think there's no doubt a little bit of hemolymph going in as well, perhaps some other tissues. But it appears for the moment, that the bulk of the diet's fat bodies. And it's funny, if you were to remove this particular tissue from the bee, you would expect to see problems like we see when Varroa are in the colonies. And it's also doubly alarming because probably some of these Varroa-induced issues can mask themselves as nutritional issues or toxicity issues, because if you're taking out this important tissue, you're not able to handle nutrition as well, or toxins as well, or whatever. So Varroa just are really bad. And then don't even forget that they're putting in these pathogens, and then you just get this double-edged sword that's just a whammy for bees. I mean, it just absolutely kills honeybees.
Can you talk a little bit about the pathogens that Varroa transmits? Yeah,
so Varroa are known to transmit at least a few viruses. The most notable of those is the deformed wing virus. And the beauty of talking about viruses is that, at least in the bee world, the viruses, yeah. You can often know what they do based on their name. So deformed wing virus, of course, results in deformed wings of adult bees that have been parasitized on by Varroa when they were young. So Varroa will invade immature bee cells, these larval cells. When the cells are capped over, they'll feed on the pupae. They'll transmit the pathogen. And since the pupae are the ones developing the wings right? These adults are growing
Uh-huh.
inside the shell of the pupa. The wings become malformed. The adult bee is born with these shriveled wings. But it's funny, the deformed wing is really only the most visible manifestation of the virus. The virus also shortens lifespan, causes behavioral issues. There's just this kind of cascade of impacts that this particular virus brings out in bees. And what I usually tell people, when you have the varroa without the viruses, you've kind of got this minor nuisance. When you have the viruses without the varroa, you've got this minor nuisance. But when you have the viruses with the varroa, you have dead bees. And that's how well they work together to impact our bees. And just to put this in perspective, and if our listeners have ever seen me give a talk about varroa, they've heard this analogy. But if you were to scale a honeybee up to the size of a human, the varroa would be somewhere in the neighborhood of a softball to a volleyball-sized critter. So imagine sticking a softball to a volleyball-sized tick on your body, and you get this image of the size of a varroa to the bee. It's really small to the naked eye, but compared to its host, it's one of the largest known parasites on planet Earth.
That's pretty crazy. So, I mean, what we recommend is to monitor, right? So,
absolutely, you have to monitor for this pest.
What are the different ways that people can do to monitor varroa?
Yeah, so there's a few different things. You can put in sticky boards, which are these pieces of cardboard that go underneath the hive, and if they're covered in a sticky coating, and varroa will rain down on those. Those have pros and cons. I much prefer to suggest alcohol washes or powdered sugar shakes. They both work on a very similar premise. You put about 300 adult bees in roughly the size of a quart jar. Then you pour in alcohol or powdered sugar. If you do the powdered sugar, you put in about 2 tablespoons of powdered sugar. In the powdered sugar case, you let the bees tumble over themselves for about 2 minutes, and you replace that jar lid with screen mesh. So after those 2 minutes, you invert it and lightly shake out the powdered sugar and the varroa, and you can count the varroa. The alcohol wash is based similar premise. You put the alcohol in there, you shake the bees for 30 seconds, and then you try to wash off and count the varroa. The magic here is we are trying to estimate a varroa to adult bee ratio, and that magic number seems to be somewhere around 3. So there's a slightly sliding scale, but it's easiest to teach it from the 3 varroa per 100 bee perspective. So an infestation rate of about 3%. So 3 varroa per 100 bees. Now, if you're doing 300 bees in an alcohol wash or in a powdered sugar shake, you're needing to see 9 or more mites before you do something about it. And the powdered sugar shake is great because you don't kill the bees, but you also can't count the bees because you release them back into the hive. So you get a kind of a loose approximation of the varroa to adult bee ratio. Of course, the downside of the alcohol wash is you kill the bees, but the upside is you can count the varroa and the bees and get a more accurate varroa to bee ratio. So a lot of commercial beekeepers will monitor for this in the field, and they'll make treatment decisions based on that number 3. If they have 0 or 1 mites per 100 bees, they won't do much. If they have 2 mites per 100 bees, they're kind of on high alert. 3 mites per 100 bee, they'll start treating. So when we talk about treatment, people are going to say, well, do I have to use chemicals? Can I use chemicals, et cetera? Well, the purpose of not using chemicals is to keep those mite loads below 3. So the idea is that not using chemicals, using resistant stock or screen bottom boards or drone brood removal, et cetera, you're not keeping those mite numbers below 3 forever. You're just delaying the time it takes for them to reach 3. And once they reach 3, you are able to interject with some sort of chemical response, Apivar, Apistan, Apigard, whatever is your treatment of choice. And that's kind of the overall recommendation. Monitor. If you see 3 mites per 100 bees, then you need to treat. You need to do something or your bees are going to suffer.
Sure. And we're definitely going to get further into varroa and more practical segments here in the future. So I hope that's okay. And we'll move on.
I know what we're going to do though, Amy. If we leave, we're going to, people are going to be hungry for more varroa. So what I'm going to do, instead of going into any more detail, I know we're going to save a lot of this for future talks. What I will say is, if you guys listening to me, Google search for Honeybee Health Coalition varroa. Honeybee Health Coalition Varroa. If you Google search that term, Honeybee Health Coalition Varroa, you'll be able to go to the Honeybee Health Coalition's page on Varroa, where they have a PDF that explains in great, simple detail how to monitor and treat for Varroa. They'll have videos showing how to do this. They'll have decision support tools, and they even have a presentation that you can download and show at your local bee club about monitoring for and treating for Varroa. So I know a lot of people don't want to be left hanging. So what I would recommend to you guys is Google Honeybee Health Coalition Varroa. You'll see this wealth of resources, and we'll make sure and link that site in today's show notes for this particular segment so that you guys can find it easy.
I always crack up because I tell people to Google things, right? And of course, we're here to provide information, but I feel like part of our job is to help people find the most accurate scientific information. You know, like we're not just telling you to go Google honeybee.
Amy, it's funny.
Oh my gosh, I
it's
can't imagine.
It's so crazy that you say
find.
that. I can't answer about most of the questions. You thought I was going to say about half the questions. I can't answer about most of the questions that beekeepers ask me. So do you know how I have to find
You're
the
just
answer?
really good at Googling.
I have to Google. So I always tell myself, if I could just teach beekeepers to Google, then I'd never have to answer questions anymore.
Yeah. And I mean, it's not just teaching them how to put in google.com and writing honeybee. It's actually finding accurate, scientifically based information, because that information is available out there. It's just a matter of being able to find it. Right?
And we need to do another segment on that. I've been giving a talk recently. No, I've been giving a talk recently on online resources for beekeepers. And I go through the 5 or 6 websites that I kind of fully endorse. Maybe we ought to save that for a future segment and get the beekeepers excited about listening to that.
Definitely. We'll teach them how to think critically on google.com.
or at least let Google think critically for them.
Okay, that's fair. All right, so we have 2 more things to talk about, nutrition and queen. So let's talk about nutrition. You know, what are we focusing on when we talk about nutrition? All I know is that honeybees need to be fed, and most people feed them sugar water. Why are we worried about nutrition?
Yeah, it's another series of good questions. So let me just tell the listeners the reason we're talking about Varroa nutrition and queens in the first place is when you look at the Bee Informed Partnership surveys talking about yearly loss rates of honeybees, and they ask those beekeepers who are completing the surveys, what are the biggest stressors that your bees face? Always, always, always in the top 5 include Varroa, nutrition, and queens. And there's a couple of others that we could talk about later. But the point is beekeepers themselves are saying these are the big issues that we face, Varroa, nutrition, and queens. That's why we harp on it. Mm-hmm. So when we talk about nutrition, the surveys, the Bee Informed Partnership surveys, almost always talk about it from a perspective of that they use the word starvation often. And starvation usually implies a lack of carbohydrates, which we know as beekeepers to mean that they don't have enough honey stores. But I like to discuss nutrition from a broader perspective and not just from the carbohydrates. So let's think about this critically. Essentially, bees forage for 2, I'll call them foodstuffs. They collect nectar that they bring back to the hive and convert to honey. Mm-hmm. Honey's not really a food per se. It's really just energy. It's liquid energy for the bees. Bees consume honey to move, to do the things that they need to do.
It's like a 5-hour energy shot.
Exactly, except it's a lifespan energy drink. Imagine if all we ate was honey.
Oh my gosh.
Or adult humans, I'm sorry.
Adult bees.
If we became adult bees, I guess all we would eat was honey. But anyway, so they eat honey to move. It's their energy source. So when we talk about, nutrition from the carbohydrate perspective, what we're having to do is ensure that they have enough honey or honey substitute. So in this case, we don't extract all their honey. I've got this long-standing tradition in my own management system is I just leave a medium super full of honey on their hive year-round. When honey flow starts, all the supers I put on above that medium super are mine, but that one medium super is theirs, and they always fill it first. So, they get the first dibs, and then I get everything I can manage them to make above that. But if they exhaust that throughout the year, beekeepers usually try to respond to that by feeding either sugar syrup, with granulated sugar mixed into water, or they feed high fructose corn syrup. And I'm not going to get in necessarily to the pros and cons of both, but essentially what the beekeepers are trying to do is replace that carbohydrate deficiency that the bees have if they have no honey. And I know this sounds harsh to the outside world, and a lot of beekeepers, some beekeepers question this practice, but in reality, the research suggests it's just fine for the bees. That bees just need sugar to move. And
they're not going to get diabetes.
No, not a bee tease. Is that what you said?
Yeah,
Anyway.
that's exactly what I said.
Anyway, so it looks like feeding sugar syrup and corn syrup is okay for the bees. Now, there's the other half of the nutrition paradigm as well, which is what most people usually mean when they say nutrition, which is pollen deficiencies. So bees collect pollen, bring it back to the hive, convert it to bee bread, and will feed bee bread, little bits of bee bread they'll mix little bits of bee bread into the larval diet. And the nurse bees, the younger adult bees, will eat it as well to mature the glands that produce the food that they feed to the immatures. So pollen is important. It's their protein, vitamin, mineral, nutrient source. So pollen deficiencies are usually addressed by beekeepers with pollen supplements. They're trying to supplement the available pollen in the environment. We often call these things pollen patties, right? These little pancake-sized things that we throw into hives. Now, a lot of beekeepers might get angry with me at this statement, but if you look at the literature on pollen patties, it's incredibly mixed. You would think the fact that a lot of commercial beekeepers do it, a lot of the time, you would think that there would be better literature support for these things, that universally the studies would show that if you put patties in hives, the bees are going to improve. And that's not the case. Emily Nordyke, who just finished her master's degree in our lab, she did projects on pollen subs, and she did a comprehensive literature review that we hope to be publishing relatively soon. And the literature's incredibly mixed. It's, as many studies say.
Is her literature review like 2 pages long?
Oh, I think
I'm just kidding,
Amy, it's probably 50 or 60. She also made like an incredibly massive series of tables where any document that's ever been published on a pollen patty studies in. So you listeners
That's
need to stay tuned. It's going to be a great resource for beekeepers. But the point I'll make is that at least half of the studies showed no impact of feeding pollen subs at all. But this is the industry-wide practice of doing it.
Yeah, it's just common knowledge for them.
It is. But again, I've been shouted off of the stage by beekeepers who say it saves their colonies, and I'm not suggesting it doesn't. I'm just suggesting
shouted off the stage, Jamie.
Well, I hate to give an example, but I think even making that statement cost me a job one time, and I'm not going to go into any more details. I'm obviously happy to be at UF, and that was decades ago. But nevertheless, the point is that I've had people, tell me that it's just not possible. Pollen subs save my hives. And that may be the case, but what we suggest is that it's very situational, number one. And number two, the greatest improvement that can be made in bee nutrition is the development of pollen subs that bees very much use and very much benefit from using. And I just don't know that we're there yet.
I mean, it's just like human nutrition, right? I mean, there's always science going on to see what's better for us and what, works well for us or not.
Sure. Absolutely. And I mean, if you think about it, there's so little we know about honeybee nutrition, and it's really an exploding science right now. And to complicate matters, even more, there's a handful of commercially available pollen subs, but a lot of commercial beekeepers just mix their own formulas, which may or may not work. And if there's no formal studies on it, it's hard to know if it's beneficial at all. And of course, a lot of our listeners will probably get angry with me, and you'll post about all the ways that pollen subs have helped your colonies. And I'm not denying it. What I am telling you in return is that it's just not universally true. It seems to be very situational. They benefit under certain circumstances, but not others. So the listeners are hearing me say this, and they're going, well, Jamie, the beekeepers through the Bee Informed Partnership saying nutrition's a big deal for bees. And you just told us, that we don't know if about pollen sub use all the time. What should we do? You should encourage research on this topic so that the scientific community at large can try to address one of these important stressors that bees face.
Well, that was going to be my question. So what would happen if we just didn't feed them?
Well, you're asking a politically charged question. Okay. I have been keeping bees for 30 years and have never in my life fed pollen subs. And I know plenty of beekeepers who say the same thing. But I also don't manage 5, 10, 15,000 colonies. I also don't move those colonies around the country 4 or 5 different places every year. I also don't move my bees to monocultures or to, pollen deserts. So So what I would argue is that there are certainly times where they seem to work. It's just that at our research in Florida, we've not found those times. So we're trying to figure out, when they benefit beekeepers most. And it looks like, at least from our research and the ways that we've done it, they benefit most when there's truly nothing else available. Because it appears that when there, at least in our research where we've done it and how we've done it, when there's anything else at all available, we lose the effect of pollen subs altogether.
Mm-hmm.
So,
and ultimately, that's a it could be a waste of money, right?
That's right.
Yeah.
But the thing is I'm not writing it off. There's absolutely, evidence that these things work in some studies. What I am hoping that this does is it breeds a more honest discussion and also pours more research funds into these topics. Emily did a fantastic job with her research. She'll have 3 papers coming out from her thesis: the general intro, which is this huge review of all pollen sub literature. Mm-hmm. The second chapter will be where she dyed pollen subs and fed them to colonies and tried to trace them through colonies, and only the adult bees had them. The immature bees.
Jamie, all I heard you say was she died.
Yeah, no, she dyed the pollen subs. No, she's still alive. She dyed the pollen subs, traced them through colonies, and only the adult bees had them. She didn't find it in the immature bees and didn't find it in the bee bread. So at least in her study, they didn't seem to be feeding it to immature bees or even storing the stuff. It just seemed to go straight into adults. And then she did a second study where she fed bees in a pollen dearth in winter, no less, in Florida winter, for what that's worth. And she tried to estimate, if you feed this much weight of pollen patties to bees, how much mass of bees does that produce? And so these 3 papers will hopefully be in review soon. And maybe even by the time some of you are finding our podcast, they may be published. So keep checking our website or subscribe to our blog for updates on these topics.
Awesome. And of course, we'll get into that, into nutrition and starvation and into other segments. So let's move over to queens. Queen quality. Is that what we are looking at? Queen quality?
Yeah, we are. But what's funny, Amy, is that when we talk about queen quality, basically we're blaming everything that happens in a colony on the queen. She gets the credit when things go right, and she gets the blame when things go poorly. Now, I don't know when this particular episode will come out, but you and I were fortunate to be able to interview Dr. David Tarpy last week. So I know that segment will probably precede what I'm talking about now. But in his segment, in his interview, he does a lot of work on this, where he looks at queen quality parameters and tries to improve queen quality. And one of the things that I remember him saying during the interview is, oftentimes what people blame on queens is unrelated to queens at all. But nevertheless, in the surveys, beekeepers say queen quality. And Dr. Tarpy from NC State was suggesting you might be claiming queen quality, but it's often not related to the queen. It's often related to these other things, and we need to learn how to address those other things better before we blame everything on the queen. It's very common practice to say, if you have this problem in your hive, you should do this and requeen. If you have this problem, you should do this and requeen. We almost always put and requeen in as an afterthought, thinking, well, if all else fails, just change the queen. That'll probably fix it. And the case is, it won't necessarily always fix it.
Well, so what about factors that affect queen quality? I mean, when we're talking about varroa, nutrition, and queens, what I've heard is that maybe there is I don't know if this is true. You can let me know if this is true. Are there miticides that affect queen quality?
So there is some evidence from some research, Dr. Jeff Williams from Auburn and some others, other places around the world have looked at the impact of pesticide exposure while queens were developing on longevity and subsequent performance and all of these things. I even think Dr. Tarpy mentioned looking at, or at least being aware of literature where people have looked at the same thing for drones. So there is some evidence out there to suggest that pesticide exposure while immatures can have downstream impacts on queens. But I would argue that there's other things as well, right? Nutrition. I want you to think about this. Worker bees live 6 weeks in the spring and summer, right? They literally work themselves to death. They can live 6 months in fall and winter. But we are so up in arms about nutrition for an individual that lives 6 weeks during the production season. Yet the queens can live 1 to 2 years.
Mm-hmm.
So if you think about it, she has maximum exposure to nutrition issues.
Sure.
She has maximum exposure to pesticide issues. She has maximum exposure to virus issues and bacterial issues because she's the longest lived individual in the hive. So she has, if there is something to experience, she experiences it most.
Mm-hmm.
So there's lots of potential stressors that can impact the queen. And so a lot of people are beginning to look at this. People often blame, they'll say, oh gosh, my colony has a bad brood pattern, so it's the queen's fault. And, David Tarpy and colleagues have shown that that's not the case. And so usually when we talk about queen quality, we talk about egg output, we talk about the productivity of her offspring, right? Are they gentle? Do they produce a lot of honey? Do they grow strong colonies? Are they resistant to diseases and pests? And that last one's the one I'll mention, even more, this idea that, we can breed queens to produce offspring that are resistant to or tolerant of a lot of diseases and pests, even though those queens aren't widely used in our industry. There are definitely pockets of beekeepers who invest in these queens, but it's not used blanket across the industry. You go to cattle farmers and they're all talking about, they're concerned about, their breed, the best bulls, the best heifers, where do I get, these stocks from? But, in the bee world, we'll just buy a queen from anybody who can produce them. Mm-hmm. And so we need to have this demand, for quality, this disease and pest resistance, this tolerance, high productivity, et cetera, so that we can improve our stocks by first improving the whole process of producing and breeding queens.
Cool. I have so many questions about queens, but we'll talk about that in a later segment.
Oh, we'll make, our podcast is, as long as you and I are at UF, we'll be doing this podcast. So if it goes on into perpetuity, or at least until one of us retires.
I was about to say, I have at least 50 years till I retire.
I was going to say, I don't know who's first, Amy. You're younger than me, but anyway, we're going to cover these topics ad infinitum, right? These people are going to be tired of hearing us talk about queens, nutrition, and varroa. What I hope, Amy, is that in 10 years, we don't have to talk about the three of these things anymore because we've addressed them and moved on to something else, right? That people are able to address these stressors significantly and hive health has improved overall.
Sure.
That's my big desire.
Yeah. In a perfect world, everyone would just remember everything we've said and then we'd be out of a job and we would have been successful at our job by being out of a job.
I'd be okay if I got to the point where I could sit back and be out of a job because the bee industry just got better. Right.
Yeah.
And the bees were healthier. Life was good. So
that's great. I remember we were talking to Jennifer Serrata and she said, if you want me to stop talking about varroa, well, let's fix the varroa problem. Right. So
yeah,
I kind of agree with that.
I'll make a political statement, but if we all, here's just my feeling, and I'm going to preach this increasingly, in the coming months at the talks I've been invited to give and other things. But if varroa is really the problem and the viruses they carry are really the biggest issues that bees face, then in my opinion, every research lab on honeybees in North America should have some sort of varroa control program. We should all come together and work with beekeepers to stamp this thing out. In my opinion, we have the knowledge and expertise collectively to do this. So here's my call to arms. Let's get together and let's do this thing. Let's defeat varroa. So anyway, I could go on and on as an impassioned Braveheart-esque speech, but I'm going to stop there because I'm not going to be as good as that speech, and neither do I wear a kilt. So people wouldn't believe me probably. So.
Oh, all right. Well, so those are just, general conversation about varroa, nutrition, and queens. This is not the first time we've talked about this. This is not the last time we'll be speaking on it. There's a lot to discuss, lots of questions from our listeners. So keep the questions coming. That way we'll find more topics and segments to discuss with you all.
It's everybody's favorite game show, Stump the Chump.
All right, we have question and answers for today. We've got something about honey, something about bees, something about honeybees,
So
a
would
couple
you
things.
expect that kind of from a question and answer about beekeeping topics?
Maybe. I don't know. I mean, we've talked about other things
Maybe
in this
raccoons or
podcast.
possums?
Yeah, sure. So we've talked about ants and other things, plants,
That's right.
et cetera, et cetera. All
We
right.
have.
So I've got 3 questions. And the first question we have is that this person had harvested honey and there are a lot of air bubbles in their honey. Is this okay? And do they need to wait for the bubbles to go away before they put the lid on?
So I would definitely encourage allowing honey to settle before you bottle it. So let me just start from the beginning. That was the short answer. So here's the deal. It is possible in the extraction process for honey to acquire a lot of bubbles, especially if it has to drop, right? It's falling from the extractor into a bucket or going through some sort of filter and into a bucket, which is why you really want it to go to a settling tank first. So what you would do is as honey is extracted, you would move it into that settling tank and allow it to sit for a couple of weeks for those bubbles to go to the top, and then you will bottle off of the bottom. If you put it straight into the bottle with all those bubbles, what you're going to get is this kind of bubbly scum across the top of all of your honey. And as your consumers open up the lids of your jars of honey, they're going to see that scuzz. And even though it's perfectly okay scuzz, they're going to be grossed out by it. So what I do, especially if this is a question from a hobbyist beekeeper, I would kind of extract into a 5-gallon bucket and then, tote the 5-gallon bucket over to a settling tank. Settling tanks are really good investments. Make sure it's food grade. You keep it on a stand. When I produced a lot of honey in my youthful days, I actually kept a settling tank in my parents' carport so it was warm. So the settling process happened faster. And so I would always bottle off of the bottom of that settling tank and got really good and clear honey. So that's my advice.
Yeah. And we also have the Honey Judge program through UF. So that, some of the classes that we offer through the university is, to help with the quality of some of the value-added products of what you all are harvesting.
That's a good point, Amy.
Yeah. I'm going to go ahead and put that shameless plug in. Yeah.
And my guess is I hate to give it away too much, but we've moved so much content online anyway. I wouldn't be surprised if very soon we didn't have a Honey Judge online component where people can learn all about handling bubbles, et cetera, in that process as well, or in that program as well.
A whole class about bubbles. All right. So the second question is, what is the best way to raise a hive? What kind of stand is the best? I've actually seen some pretty creative stands, so I'm excited to hear your answer.
It's a good point, Amy. The deal is that there's about 1,000,001 different types of stands. And so really, to me, there's a couple of key things to consider when stands. Number one, I do not like to bend over for anything.
Sure.
And that's the older you get.
How do you tie your shoe?
Yeah, well, that's why I have kids.
Do you have Velcro? Do you have Velcro shoes?
No, but that's why I have children. That's like children time. No.
Oh my goodness.
In all seriousness, I don't like to bend over while I'm working bees, especially if you're picking up boxes or moving boxes. So what I like is when I get to my brood box, I like the brood box to be naturally about waist height. So the top of the brood box to be somewhere, right at or just below the belt on me, right? So I'm not having to bend over to work it. So what I have discovered is if I use 2 of those very standard cement blocks, so one stacked on the other, and then I have a 4x4 that runs from 2 cement blocks to another 2 cement blocks. So basically the cement blocks are spaced 7 feet apart. So 2 on one side, 2 on the other. And then I have 2 4x4 runners, going from one pair of cement blocks to the other, that raises the brood box to the level where I don't have to bend over. So why then would I make it 7 feet long? Well, I keep one colony on one side of that stand, one colony on the other side of that stand, and the space in the middle, I can put the lids and the supers on as I'm working a particular hive. So if I'm working hive 1, I'll take the lid off, I'll turn it over onto the 4x4 space between the 2 hives, and then I'll put the supers on that lid and more supers on that lid until I get to the brood box and work the brood box, etc. So I know it's kind of a little difficult to describe that way. There are some documents that we have that kind of show this setup. They're not documents about setting up hive stands. They're just documents that you can see the hive stands that we use. Maybe we can link to a few of those.
Sure. But
that's just a very simple one. It just basically takes 4 cement blocks per 2 colonies with 7-foot 4x4 runners. So again, 2 cement blocks on one side, 2 cement blocks 7 foot away, and I put the runners on top. Now, with that said, I've seen people do the same thing, but rather than use 4x4 runners, they use ladders, like step ladders.
Mm-hmm.
I've seen people only put them on cement blocks. But I think the real key is not what you're putting them on. It's how high off of the ground that you're keeping them to make it convenient for you. And that's kind of what I shoot for when I'm working my bees.
Yeah, when I first started beekeeping, we were friends with this arborist, and he had all these huge tree stumps, and they were perfect. They actually worked out really well as far as putting a hive on the tree stumps. So they were
that's super cool. And one of the things I'll say, Amy, is that when I'm thinking about hive stands, I'm trying to think about things I don't have to replace.
Mm-hmm.
And the beauty of using cement blocks is you won't have to replace them unless you break them. And 4x4, you can use the treated 4x4s and they'll last for a very long time as hive stands. So yeah, when I think about tree stumps, I would worry, I suppose, over time about rot. But at the end of the day, you really just want those colonies up off the ground where it's convenient for you. With that caveat, obviously, most commercial beekeepers work their colonies on pallets, like, on a pallet, super, super thin. So you're 4 or 5 inches from the ground, 6 inches from the ground. But that's what they have to do because they work so many bees and have to move them around.
Yeah. All right. So the last question is, would it be okay if I got rid of deeps and used something smaller? Are there pros and cons to using mediums only?
It would be more than just okay. Here's what I say. There's some basic biology facts that you have to know about bees to keep bees. Almost all, I'll say 90%, maybe be a little bit more conservative, say 80%, at least 80% of the management is opinion. Whether or not you use excluders, whether or not you use deeps, whether or not you use mediums, whether or not you use 8-frame colonies or 10-frame colonies. It really doesn't matter. The bees don't care. So really all of that stuff is stuff that's up to you. So historically, the colony style I use is a deep brood box, 10 frames, a metal queen excluder, and then a medium 10-frame super. That's what I use. But I know plenty of people who use exclusively mediums because it's easier to pick up mediums than it is to pick up deeps. For the same reason, I know people who use exclusively shallows, just a whole hive made of shallows. And that's also fine. I would argue, though, the only drawback to using mediums and shallows is you have to buy more boxes, and they're not priced based on their size really well. It's not like a shallow's half the price of a deep. It's still fairly expensive, so there's a little bit more cost. And the other drawback that I would see to them is that when I have to look for a queen, for example, I'm looking among 10 frames in a deep brood box. But if I'm having to use 2 shallow supers to make up one equivalent of a deep brood box, now I'm looking through 20 frames for a queen. So there's just a few oddities like that you'll find that the fewer boxes you have to use, the easier it is in those things. But the fewer boxes you have to use, usually the heavier the boxes. So but the long answer, the short answer is it really doesn't matter. Feel free to experiment with hives composed exclusively of mediums. Lots of people like them. You can even experiment with 8-frame mediums or 8-frame deeps, whatever. All of that stuff is just personal preference. The bees don't care at all.
So there's no one hive fits all.
That is right. And in fact, Amy, we have a document somewhere that maybe we can put in the show notes. It talks about hive choice and configuration, where I wrote specifically about this topic. What are the different configurations and what pros and cons are associated with each? And so all of that stuff's covered in that document. Maybe we can make sure and link it.
Awesome. Yeah. So, and so thank you for that, for all of the questions. And audience, I hope you all are keeping your questions and sending them in. I am receiving some questions, but we need more. So
absolutely. Yeah. Make sure you go to your favorite podcast platform or rate us so we can spread the news. And while you're there, make sure you drop us some comments and questions. We appreciate your listenership.
Hi, everyone. Thank you so much for listening to this week's episode of Two Bees in a Podcast. We would like to give an extra special thank you to our audio engineer, James Weaver, and to our podcast coordinator, Jacqueline INJ. Without their hard work, Two Bees in a Podcast would not be possible.
For more information and additional resources for today's episode, don't forget to visit the UF-IFAS Honeybee Research and Extension Laboratory's website, ufhoneybee.com. Do you have questions you won't answer it on air? If so, email them to honeybee@ifas.ufl.edu or message us on Twitter, Instagram, or Facebook at UF Honeybee Lab. While there, don't forget to follow us. Thank you for listening to Two Bees in a Podcast.